study biology and environmental science, and I'm really interested in either being a field researcher or getting involved in policy and more management and conservation.
Hi, my name's Sophia. I'm a health education major, and my career aspirations are to become a health coach or a naturopath doctor.
Hi, my name's Bree. I'm majoring in biology and I'm hoping to become a physician assistant.
Conducting surveys is an important part of field research. Determining species composition and abundance is really fundamental to ecology. So, because of that, it's important to make sure your surveys are comprehensive, accurate, and cost-effective. There are a ton of ways to conduct surveys that differ depending on your research question and target organism. So we're just going to focus on outlining some of the basic and most widely used techniques for invertebrates, birds, mammals, reptiles, and frogs. Let's start with everyone's favorite, mammals. Live trapping is a commonly used method for surveys of small to medium-sized mammals, ranging up to around the size of a wombat. The main methods for live capture of mammals are Elliott cages, wire cages, and pitfall traps. Elliott traps are small rectangular aluminum traps. You put a food bait in the back, and when an animal enters, it triggers a foot pedal which closes it behind it and prevents the animal from leaving. There are many variations on that basic design, ranging up to larger wire cages which are used for bigger mammals. Elliott traps are usually only used for mammals up to the size of rodents. Pitfall traps are also frequently used. They're made by digging 2 holes for PVC pipes, which you embed in the ground so the top of the pipe is just at or below the surface. They're especially effective when you place a drift fence between the 2 pipes. That's usually made of mesh or some other plastic. So the animal's running along, and when it hits the fence, it'll run alongside the fence and then get stuck in the pitfall trap. It's important to consider that if it's going to rain over the time of your survey, you should place a little flotation device inside the PVC so that the animal has a refuge if it starts to flood. An alternative to live capture is observation. One of the main methods of direct observation is spotlighting. As anyone who's driven at night and seen eyes glowing in their headlights knows, animals' eyes produce eye shine when they're hit with an external light source at night. So spotlighting is basically walking your transects, shining your light source, and looking for eyes to ID. Those are especially effective in open habitats and when you're looking for arboreal or nocturnal critters. Another commonly used method of observation is camera traps. Camera traps are like they sound, just small cameras which you place out in the field that are motion activated and then capture an image of whatever organism triggered them. With camera traps, it's important to know a bit about where your animal will be so you can correctly place the traps and get the right height for the animal. Those are really effective because they reduce human labor costs and because they avoid the potential problem of a human in the field disturbing the predators, the mammal, and scaring them away.
Moving on to birds. The best method of live capture birds is the use of mist nets. Mist nets are simply a strip of mesh hung up between 2 poles. When birds fly into the net, they get trapped and they can be collected by observers. Mist nets are frequently used for tagging and tracking as well as mark recapture. Observation is also used for bird surveys. Observation is based off both visual observation and call ID. This is usually done by walking transects or point counts. Point counts involve standing at a point for a given interval and noting any observed species. Transects are essentially the same, but observations are taken while walking a set distance. Calls can also be stimulated by call playback.
Invertebrates are a diverse taxa and there are a wide range of survey techniques. For larger invertebrates such as bees and butterflies, direct observation can be an effective method of surveying. However, invertebrates are well hidden and more active searches are needed. Hand searches can be used focusing on suitable habitats. Many light-flying invertebrates are attracted to light and can be captured by simply placing a white sheet in front of the light source. The insects will fly onto the sheet and can be collected there. Lace traps are tent-like mesh structures which taper to a narrow point at the top where insects are collected and exposed to a chemical which kills them. Sticky traps are simply sticky traps, which immobilize vertebrates who walk over them.
With many small reptiles, catching is the best way to get an accurate ID. One familiar technique is simply diurnal hand searches, where the researcher manually searches suitable microhabitats such as under logs or in leaf litter, where reptiles may hide out during the heat of the day. Another technique is spotlighting, especially in the hours right after sunset. Traps can also be used. We've mentioned some which are pretty effective, like pitfall and Elliott traps. Also, funnel traps can be very effective at capturing snake species which overlook others. So how that works is you just have a tapered funnel and the snake can enter but then gets trapped and can't exit.
The best methods for frog capture are similar to those we have seen before. Pitfall traps are often used for adult capture. Funnel traps placed in aquatic environments can be used to capture tadpoles. Hand searchers are also possible, however, this should only be done when absolutely necessary, and great care must be taken when handling frogs as they are very susceptible to disease transmission and common products such as bug repellent and sunscreen. Visual and auditory detection are also good options for frog detection. Frogs have very distinct calls and can be identified in that manner. Similarly with birds, this can be done by walking a transect, or more commonly a quadrant with frogs, or by point counts.
It is also important to make sure that animals are being trapped in an ethical way. Some ethical trapping methods include
trapping the animal without harming them. This can be done by handling the animals with care. For example, creating a trap for a fox that still allows the animal to eat is better. Just making sure you're taking into account the animal's basic needs.
Reducing the likelihood of predation. Traps make species vulnerable to attack, so to avoid this, traps should be checked frequently.
washing hands and equipment between handling to prevent the spread of disease.
There should also be minimal to no trapping of non-target animals. This can be done by creating traps with holes in them that filter out smaller, untargeted animals.
Researchers should perform a quick assessment and quickly return animals back into the wild. Time is important because animals caught by researchers must be released from a trap within 24 hours according to the Australian Wildlife Conservation Guidelines.
The field researchers should also take into account how their trapping techniques will alter their animals' habitats and minimize the disruption of normal activities for the individual animals. One example of ethically trapping animals was reported by two scientists in Australia in 2007, who successfully trapped pygmy possums without causing any harm to the animals. This was done by using shallow traps, which eliminated the amount of drift net and decreased likelihood of non-targeted mice. In doing so, they minimized the amount of stress inflicted on the possums and were still able to successfully collect the data.
Collecting thorough and accurate data is an extremely important aspect of data collection. Errors in data collection occur when researchers do not have specific goals in the information they are trying to acquire. Mistakes can arise when there is a lack of knowledge about the collection process, vague data measurements, a lack of attention towards reviewing the data, and not having a regular analysis of the data. In order to collect the most accurate data with the least amount of errors, I have comprised a few steps for the best ways to collect field data.
First, completeness. Making sure that all the data that you need was collected. If there's an error involved in incomplete data collection, it can lead to inaccurate results.
Consistency, constantly being aware of the data collected and making sure that nothing is too far-fetched from what is expected.
Timeliness. Collecting data during the proper time interval can be crucial to collecting accurate data.
Validity. After collecting the data, it's important to review it and make sure it's all valid and matches up.
Accuracy, reviewing the data to make sure that all the information collected was exactly what was measured. Using inaccurate information to convey the data is extremely misleading, and all reasonable steps must be taken to prevent this.
I'm going to walk through how to ID one of my favorite organisms, ants. Anyone who lives on campus will have seen these cute little ants with their distinctive green butts, or properly called gasters. I used Stephen Shattuck's book, Australian Ants, Their Biology and Identification, to identify the ant species I was curious about. To identify the species, one of the most important things to note is the color and shape of the gaster, or rear section. The ants I'm interested in have smooth gasters attached to the middle section of the body by a single segment. That tells us it's part of the subfamily Formicinae. From here, we can use the color of the gaster to identify the species. The only ant in this region that has such a green, elongated gaster is the Ocelophila smaragdina, or commonly known as weaver ant. If we were to look closer, these ants would also be identifiable by a large number of mandibular teeth and very few palp segments, palps being structures on the head of the ant used to detect pheromones. For these ants, you can take a shortcut because it is easy to see them walking to and from the nests they create in trees and shrubs. These nests are formed with silk produced by their larvae. How cool is that?
One of my favorite animals to run into on campus is the bush turkey. According to the Australian Birds Guide, this is a common species in North Queensland, Australia, especially in the drier, scrubbier areas. Identifying this species is fairly easy because they are not afraid of humans and are very common. According to the Queensland Nature Conservation, the bush turkey is listed as least concerned in conservation and a low priority. Another reason why they are easy to identify is because they have a bright red head and neck with a long yellow wattle that hangs over from the neck. The bush turkey has long, thin legs with strong, stable feet. The average bush turkey is about 60 to 70 centimeters long. If you see a bush turkey in the wild, they will most likely be scavenging for food by raking up leaf litter with their large feet. They like to feed on nuts, seeds, fallen fruit, and insects.
Now we are going to talk about how to correctly identify turtle species found in the Ross River. According to the Queensland East Coast Freshwater Turtle Identification Guide, the first step is to see if the turtle's forelimbs have 4 or 5 claws. This specific turtle has 5 claws. The second step is to look at whether or not the turtle has a nuchal scute on the front of the marginal portion of its shell. In this case, the turtle has a nuchal on the top and 24 marginal scutes. Next would be to identify the scutes on the bottom of the shell. If we look specifically at the gular, intergular, and pectoral scutes, we can tell that the turtle has a very similar shape to the image on the right. Both gulars are touching with the intergular scute directly underneath them. The portion of the pectoral scutes are also slanted downwards instead of upwards. By identifying the smooth, flat scales behind the eyes and the absence of pointed spikes on the neck, we can classify this turtle as a Carriff river turtle through the dichotomous key.
Thanks for watching!